Good afternoon, students. Yesterday we have discussed about induced mutations and the causes of induced mutations. So, induced mutations are those mutations which are deliberately carried out by using mutagens. A mutagen may be a chemical mutagen or a physical mutagen. Now, we discussed two chemical mutagens. Number one, base analogues, and what is the effect of base analogues, how do they cause mutation. And number two, we discussed about agents that that modify purine and pyrimidine bases, that is nitrous oxide, hydroxylamine and alkylating agents, and what is the effect of those agents on an organism. Right, now today we are going to discuss about the third type of chemical mutagen, that is agents producing distortion in DNA. So let us continue. Agents that produce distortion in DNA are a type of chemical mutagens, and they are generally intercalating agent. They are intercalating agents. Okay. What are intercalating agents? Intercalating agents are generally acridine dyes. They are fluorescent dyes like proflavin, acriflavin, acridine orange, ethidium bromide, etc. Now, these dyes are actually planar molecules. These dyes are planar molecules like that of the nitrogenous bases. And they are almost having the same size. They are not exactly same, but almost having the same size. Okay, and how do these agents cause mutation? These agents, that is intercalating agent, for example proflavin or acriflavin or acridine dyes, they get intercalated between two nitrogenous bases. That means they get sandwiched between two nitrogenous bases. That means they intercalate between nitrogenous bases. Okay. They distort the three-dimensional structure of the helix and cause single nucleotide insertion and deletion in replication. Okay. So, what do intercalating agents do? Why they are named as intercalating agent? Because they intercalate between two nitrogenous bases. They get fitted or integrated or sandwiched between two nitrogenous bases in a DNA. Okay, depending upon whether they get intercalated in the template strand or the newly synthesized strand, they bring about deletion or insertion kind of mutation. They bring about deletion or insertion kind of mutation, or you can say frame shift mutation. These insertion and deletions produce frame shift mutation. Okay. So how this happens? So this the upper one is proflavin. It's an acridine dye. You can see it's a planar molecule. Okay, somewhat similar to a nitrogenous bases, and hence it can get intercalated between two nitrogenous bases. This is an example of acridine orange. Okay, and look at this. This is the structure of DNA. Okay, so this purple color are nitrogenous bases, and you can see this. These are the, okay, wherever I am pointing, these are the intercalating agent. They are the fluorescent dyes like acridine orange or proflavin or acriflavin, and they get integrated in between. These are the two nitrogenous bases, and in between the two nitrogenous bases, this the one where I drew the line is the intercalated molecule. It is a acridine dye. This is how it gets intercalated between the nitrogenous bases. Okay, and what happens actually? Now look at the first diagram. Here you can see that there is this molecule. This is a intercalating agent. It is a acridine dye like proflavin. So if a molecule, if a intercalating agent like acridine dyes get integrated between two nitrogenous bases, say number 2 and number 3. So this dye has got intercalated between second and third base in the template strand. Okay, because it is there in the template strand during the first replication. Okay, during the first replication, at random any base will be added opposite to this acridine dye, because DNA polymerase enzyme does not recognize it as any base. So at random any base is incorporated against it. Say for example a base X dash is added against this acridine molecule. Now what happens in the next generation, what will happen? Because X dash molecule is present in the newly synthesized strand, in the next generation, okay, in the next second replication, a base X will be added, okay, against X dash, and hence the length of the DNA strand will increase. So it will lead to insertion kind of mutation. Right. Now look at the second example. This is an example of deletion kind of mutation. Okay. What happens here? The acridine molecule has got integrated. The acridine molecule has got integrated in the newly synthesized strand. Okay. So the intercalating agent is present in the newly synthesized strand in the first replication. So base complementary to 1 is added, 1 dash, 2 dash, and this intercalating agent comes across the third base. So DNA polymerase cannot recognize it what it is. So it will be left as it is because it has occupied. It does not allow, because it is present exactly in the complementary position of base number 3. None of the complementary nitrogenous base will be added because it has blocked the site of the third base. So no base will be added, and this will lead to deletion kind of mutation. So in the next generation or in the next replication, the nucleotide number 3, the base number 3 will be deleted, and the length of the DNA strand will get shortened. Okay. So, if the acridine molecule gets intercalated in the template strand, it causes insertion type of mutation. If it gets intercalated between the two bases of a newly synthesized strand, it leads to deletion kind of mutation, and hence causes and hence causes frame shift mutation. Okay. So, intercalating agent cause frame shift mutation by causing deletion or insertion of one base. Right. Okay. So now let us discuss about the second type of mutagen, that is physical mutagens. Okay. So radiations are generally the physical mutagens. Radiations, high energy ionizing radiations like X-rays or gamma rays, and UV rays, both are the radiations which are used or which actually are responsible for causing mutations. Okay. So radiations as physical mutagens. Let us discuss X-rays, gamma rays, cosmic rays, UV rays, all are capable of penetrating the tissues and damaging the DNA. Okay. What is the effect of these radiations? Whether they are high energy ionizing radiations or UV light, whatever. What do? They cause ionization of nitrogenous bases. They cause ionization. They directly damage the DNA or indirectly damage the DNA. They directly damage by causing ionization or excitation of nitrogenous bases, or indirectly by forming high energy free radicals, Okay, so they either create free radicals or they ionize the bases. Okay. Now, let us discuss about what is the effect of X-rays. X-rays are high energy ionizing radiations. Okay. Whenever a cell is exposed to any radiation, be it high energy ionizing radiation or any other radiation, what happens? The proteins, okay, the lipoproteins, the carbohydrates, all are affected. Now, if you look at what is the effect of X-rays, then X-rays actually break the phosphate diester linkage. Okay, X-rays break the phosphate sugar linkage. Because of that, breaks are created in the DNA strand. And the break in the DNA strand may be created at one or more place. And because breaks are created in the DNA strand at one or more place, DNA bases are released. Okay. So DNA bases are removed from the DNA strand because of breaking of DNA strand. So X-ray can break the DNA strand and remove the bases get removed. So this causes very harmful effect on the DNA and leads to mutation. Sometimes the break occur in one of the strand of DNA, but sometimes the break can occur in both the strand of DNA. So when breaks occur in both the strand of DNA, a part of DNA strand will be deleted or removed, and it will have a drastic effect on the characteristic of the organism, phenotype of the organism. Okay. So ionizing radiations frequently result in double stranded breaks in the DNA, the last point. Okay. So it breaks the DNA and damages the DNA. X-ray. Now, ionizing radiations like X-ray break covalent bond, including those in DNA, and is the leading cause of chromosomal mutations. Okay. Now, whether they are X-rays or whether they are UV rays, they have the ability to cause mutation and damage a cell, and hence they are also used for sterilization. Okay, they are used for sterilization. Now, let us discuss what is the effect of UV light on DNA. See, though UV light do not have high energy and they do not cause ionization of bases, but they create free radicals. Okay, right. Now both the DNA as well as RNA, when it is exposed, when a cell is exposed to UV light, both DNA as well as RNA absorb the UV light, and high energy free radicals are created. Okay, when free radicals are created, one base may get converted into another base. That means transition of bases, transition from one to another base, and a transition mutation can take place. One base may get converted into another because of creation of free radicals. Okay, if this transition type of mutation takes place in mRNA, then few proteins are affected. But if this kind of mutation transition takes place in DNA, then it can cause a huge damage to the organism. Then it can affect all the proteins that are coded by that particular DNA. Right. Now, the most important effect of UV light is the pyrimidine dimer formation. Okay. Whenever a cell is exposed to UV light, the DNA may absorb the UV light, and generally thymine dimers are formed. If in the DNA strand, if two adjacent thymine bases are present, then the two thymine molecules, with the help of their 5,6 unsaturated bond, they join each other and form a cyclobutane ring. So you can see one thymine here, another thymine here. The fifth and the sixth, the 5,6 CC bond is unsaturated, and when they absorb UV light, thymine dimer is formed, and 5,6, with the help of 5,6 unsaturated bond, it forms a cyclobutane ring. This one. Okay, so this is a cyclobutane ring. 5,6 unsaturated bond of one thymine has combined with another, and a cyclobutane ring is formed. Thymine dimer is formed. Now, this thymine dimer cannot be accommodated in the DNA, and hence it causes a distortion in the DNA. It causes a kink or a distortion in the DNA. Now, what happens? Dimerization of pyrimidine residues takes place. But when studies were carried out, it was found that thymine dimer formation is 50%, and thymine cytosine is 40%, and CC dimer formation is just 10%. So most of the time thymine dimers are formed. Now you look at this diagram and just see. This is a DNA strand, and these are the two adjacent thymine residues. Okay, when the cell absorbs UV light, when the DNA absorbs UV light, the two thymines join each other, I mean they dimerize. Okay, and that causes a distortion in the DNA, because they cannot be accommodated in the DNA strand. It forms a kink or distortion and thus damage the DNA. If this damage is not repaired, if this thymine dimer formation damage is repaired, then no problem. But if this damage is not repaired, it will lead to mutation. Okay, if the damage is not repaired, it will lead to mutation. Okay, one more thing, what happens? The nitrogenous bases absorb UV light and form okay, now nitrogenous bases absorb UV light and cause photohydrates. They absorb a water molecule. A water molecule is added to the molecules DNA bases, and photohydrates are also formed. So this is how UV light damage a DNA or cause mutation by formation of thymine dimers, mostly or thymine cytosine or cytosine dimers, and forming photohydrates. Addition of water molecule to the nitrogenous bases takes place and photohydrates are formed. This is how physical radiations cause damage to the DNA. If the damage is not repaired, it will lead to a mutation. Okay, so that's all about physical and chemical mutagens, and in the next class we will discuss about lac operon of E. coli.